sect of the Mississippi River, and go for it because I know it's going to be fantastic and interesting.
Okay. Well, thank you so much. It's an honor to get a chance to share some of this data with you. This is very preliminary, hot off the presses data. My graduate student Michael Hansen just finished analyzing some of the things that I'm about to show you today. What this is a collaborative effort between OR Northwest's transect of the Mississippi River from last year and my lab at LSU. And we worked with OR Northwest to process the samples that they were taking to examine the microbial communities in the Mississippi River. And so what I hope to do is introduce the topic of why we should care about microorganisms in rivers at all, and then walk you through some of our beginning data. So I hope that that will be clear by the end. Now this picture right here is what I like to start with a lot as a microbiologist, because I think it nicely frames a series of arguments or a series of questions that we have to tackle. This is the surface of a filter. Those little holes that you see are the holes of the filter, and this is a water sample that's been filtered onto this filter. And what you see are a really amazing collection of microorganisms that exist in the water naturally. And now these are not pathogens necessarily. Most of the organisms that occur in marine and freshwater systems are actually mostly just doing their own business involving biogeochemical cycling of important parts of environmental systems. So they're dealing with the carbon cycle, they're dealing with sulfur cycle, etc. So most of these things aren't going to infect you. But you can also see that when we look at these, there are a number of very interesting different shapes and sizes. We have the capability to determine that there's a little bit of differences, a little bit of difference between any one of these. But if you look even more closely, you'll find pretty rapidly that you might be having trouble distinguishing whether one is the same as the other, or just a slightly larger version of another, or maybe one is dividing and it hasn't quite finished yet. The other thing that this brings up is it's very hard to determine what any one of these organisms might be doing, all right? How do we know what they're eating? How do we know what kind of compounds they're producing? How do we know what their role is in the environment? And this gets even more profound when you think about the fact that this particular organism right here, for example, has the same width as your bicycle spoke cut into 10,000 slices. So that's a really tiny thing to try and identify, a tiny thing to try and understand. And you know, we want to understand them the same way that you would understand an elk or a tree or any other biological entity that's out there. We want to observe it. We want to bring it back to the lab maybe. We want to try and figure out what it's doing out there. But that's a little more challenging for organisms that exist in the water column, because not all of them can be brought into the laboratory. Some of the questions that we're trying to pursue here are, you know, what makes these microorganisms different from one another? What makes them different from organisms in other places? You know, why are these organisms here and not somewhere else? Or why are these organisms here instead of other organisms? And what exactly are they doing? And so what I'm going to show you today are our attempts to do some of this kind of work in a specific system. But before that, I want to try and convince you why you should care about microorganisms at all. So this is a graphic from a review that was published a few years ago that approaches this question from the perspective of one really important cycle, which is the carbon cycle. Turns out that microorganisms, although they are very, very tiny, are present in enormous numbers, right? So in any drop of seawater, we can have 10 to the 5th to 10 to the 7th microorganisms. And when you extrapolate that to the entire ocean or to an entire lake or to an entire river, that means that there are trillions and trillions and trillions and trillions, thousands of trillions of microorganisms total in those systems that are doing very important things. One of those is interacting with the carbon cycle. And what we have here is a graphic where we show that in a water system, light is being absorbed by photosynthetic organisms. Those are transferring all of that photosynthetic energy to fixing CO2 from the atmosphere into organic carbon. So this is just like a tree or a plant. You have the phytoplankton, which is what these microorganisms that do photosynthesis are called, taking light and turning CO2 into organic carbon. Some of that organic carbon falls into the sediment and is sequestered. Some of that organic carbon is respired, which means it's eaten and turned back into CO2 the same way you and I eat a hamburger and breathe out CO2 later. There are microorganisms that do both sides of this cycle. These are all microbial processes. And so obviously, as we are concerned with the concentration of CO2 in the atmosphere, with increasing CO2 in the atmosphere as we burn more and more fossil fuel, we're very interested in the organisms that are doing these processes and what the balance is. Is there more CO2 getting turned into organic carbon, or is there more organic carbon getting turned into CO2? These are part of the process that we really want to try and understand, even in a river system. Now, there are some other important questions about microorganisms, specifically in rivers, and I hope that by the end of this, you'll be able to see that we have, a couple of these questions partially answered. One of them is, are all microorganisms in all parts of the river the same, or do they change? Is the river basically a homogeneous system with organisms that are similar from top to bottom, or is it different? If they are different in different parts, why is that? What are they doing that might be different? Do the influences of cities and tributaries along the river alter the microbial community, the collection of organisms that are there? And what are they doing there? What are they doing? All carbon cycling type stuff? Or are they interacting with other nutrients? Are they creating toxins? What exactly are they doing there? Which ones, alluding back to the last slide, which ones are making CO2 into biomass and organic carbon, and which are doing the opposite? And so the first step to answering all these questions is really just to go out there and look and see which organisms are present, and that's what the goal of this study was. And so what I'm showing you here, a trip down the Mississippi River, this is a map of the United States, and each of those dots is a place where the rowers stopped and took samples. The rowers consisted of the Or Northwest team with Jordan, Greg, Pat, and Marcus, and I'll put all their pictures up at the end. They have rowed across the ocean a couple times. They've rowed around Vancouver Island. They've rowed more than most people, they've rowed more, each one of them, in a day than most people have ever rowed in their whole lives combined. And they were very well suited for this trip. They planned it out meticulously, and we sat down in advance and worked out a protocol for them to sample while they were going down the river. And this took a couple months, but they made it all the way from the headwaters at Lake Itasca all the way to the Gulf of Mexico. We have a really cool picture that they took of their trip with this beautiful old steamboat and one of the rowboats. You can get a sense of how small those boats were in the foreground. I told you that we developed a very simple filtration protocol for them. This is a little bit blurrier than I hoped it would be, but you can kind of get the idea. It's very simple. It was handwritten. The rowers would take a sample of water from the side of the boat. We would use a large 60 ml syringe where they would attach a couple of filters. They would push the water through the filters a couple of times to collect biomass, and we would also collect the flow through for nutrient analysis. The important things to remember for the purposes of the data I'm about to show you is we had a filter up here that was 2.7 microns allowed things smaller than 2.7 microns through. So this will trap large microorganisms and particle-associated microorganisms. And we also had a filter that trapped much smaller organisms, a 0.2 micron filter, which is about the same size as the filter I already showed you with the organisms on it. We also had flow through that was collected for nutrient analysis. So this is things like nitrate and phosphate, nitrite, ammonium, and some other nutrients that were in the water. The rowers took these samples. They put them into coolers that we monitored for temperature, and at various predetermined sites, they would ship off a cooler full of samples, send it to LSU. We would process them, and at that same site, an empty cooler would be waiting for them with new supplies that they could pick up for the rest of their journey. So it was a pretty cool system. It worked out pretty well. We didn't have any major snafus. This is a picture of Jordan and Greg sampling. You can get a sense of the scale of the syringes and the water that they were holding. They're both wearing their gloves, which is excellent. We have a little video protocol on the Order Northwest website that we developed to teach them how to do this, that everyone can use. And so I want to go back to looking at this chart because there's a few more things on here I need to point out. The blue dots are what are consistently referred to as the geographic upper Mississippi River. And this goes all the way from the headwaters above the Twin Cities all the way down to basically where the Missouri River comes in at St. Louis. Then people have referred to the green dot area as the Middle Mississippi River, and that's between the Missouri and the Ohio River. And then below that is considered the Lower Mississippi River. And so we've used the traditional definitions to colorize this map, and those are the same colors that you're going to see in the remaining graphs that I show you. Really, the Missouri to Ohio River section is less than 200 miles long, and although it's a very important segment in terms of shipping and commerce, it's much smaller than the other two. And I think I'm going to hopefully convince you that maybe it's a lot more like the Lower Mississippi River than the upper. I believe the very first sampling points right here are just above Minneapolis for your reference point for those of you who know the Upper Mississippi River well. Okay, so I want to go back to this slide and talk a little bit about how we determine what these organisms are and what they might be doing by looking here at this filter one more time. These microbes are on the surface of the filter, and so this is kind of what it would look like if you were to zoom in on the 0.2 micron filter, the second filter that I pointed out in our protocol. And we can learn a lot about these organisms by looking inside their DNA. And so that's the way that we've chosen to identify who these organisms are and what they're doing. So we take the filter and we mash these cells up, and then we do special chemical extractions of the DNA inside them. And then we can take advantage of a molecular fingerprint that all organisms have inside their genomes. This is a picture of a 16S ribosomal RNA. This is the secondary structure. It's pretty fancy looking. But what this does is it curls up inside the ribosomal proteins, and it helps the organism translate RNA into protein. Every organism has a 16S or an 18S rRNA gene in its DNA. And so we use that gene for this particular RNA as a DNA fingerprint to identify organisms and classify them and sort of put them into different categories. So here's some of the data from our trip. Each one of these plots is showing a bar chart of all the organisms or all the sites along the x-axis. And the ones on the left are the free-living organisms. The ones on the right are those that were trapped on the larger filter, the particle-associated organisms. And what this is doing is classifying organisms based on their classification at sort of a high taxonomic level. So this is based on the class level of the 16S rRNA gene. And really what I want to point out to you here, because the labels are unfortunately a little hard to read, is that the colors are slightly different, and they're definitely different in different shapes and forms. And you can see down here at the bottom, this lower pink level at the very lowest level is a little bit lower in abundance than the one on the right. You can also see if you go 2 layers up that there is a much more patchy distribution of that third layer of colors than there is on the left. And so what I'm really getting across here in a broad definition is that we are seeing alterations between the free-living and the particle-associated organisms when we try to identify which organisms specifically were in our sample. And it looks like there's some significant differences between those that are trapped on the particles or the larger organisms and the ones that came through that big filter and are on the very smallest filter. We can look at this a different way with what's called a non-metric multidimensional scaling plot. Now I want to take a couple minutes to just explain what this is, because this may be unfamiliar to some of you who are watching. Essentially what each one of these dots are a a complete metric of the entire microbial community at one site. All the ones that are circular, kind of all the ones that are on the right, are the communities from each site that we collected on the smallest filter, the 0.2 micron filter. And all the ones on the left in the triangles that are collected were those that were collected, most of them are triangles, were collected on the larger filter, the 2.7 micron filter. Now the way that this works is these communities are separated.
Cameron?
Yes, sir. Yes, sir.
There is a question there from John Sullivan. What defines free living, I believe it is?
Yeah, that's a good question. This is sort of an operational definition. In our system, we're defining it as anything that goes through a 2.7 micron filter, because most things that are attached to other things will get collected on a filter of that size. But that's an operational definition, and you can use different types of filter sizes to restrict that level more or less. Some people use 1.8 micron filters or 0.8 micron filters. Some people use 4 micron filters or 0.4 micron filters. So there's a different, these are just choices you have to make to sort of size fractionate these organisms. But in general, what a free living organism means is it's an organism that's floating freely in the water column, and it is not attached to something else, not associated with the surface of a larger organism or a piece of dirt or a mineral that's floating in the water column. Okay. So what I want to show you here, though, is that each one of these dots essentially represents an entire community, and the dots are separated based on how different the whole community is. And the analogy for this is sort of like how you could tell that a forest is different than a desert very rapidly as a human by looking at the types of trees, plants, animals that are present there. And you would know intuitively that a desert has very different plants and animals by observation and by thinking about it compared to a forest. Well, a forest and a desert would be very far apart on an NMDS plot like this. And so the things that are more similar in their composition of different groups of organisms are closer together on a plot like this. And one of the main patterns that comes out of this, again, these are colorized according to upper, middle, and lower on the map. They're also shape defined by circle and triangle, is that you can kind of draw a line down the middle here that nicely separates the free living and the particle associated organisms, or the ones that are on the smallest filter fraction and the larger filter fraction. This is a common pattern. It's been seen in a lot of systems. It's something that we would probably expect. And so it's not terribly surprising at this point, but it is a piece of data that is worth noting because we're seeing very different communities between the particle associated and the free living fractions. Okay, so to continue, I want to come back to where we sampled, and let's look at the Missouri River and the Ohio River coming in here. And I want to point this out because these are where the color transitions happen. From blue to green is where the Missouri River comes in, and the Ohio is from green to red. And now Jordan and his team were sampling both above and below all the confluences of the major tributaries and the cities. So we do have some samples from directly above the Missouri River confluence and directly below it, as well as the same type of arrangement for Ohio and many other rivers. One thing that also pops out on this plot is that there is a very strong separation between the communities that are in the green and red, with one small exception up there in the upper left corner, and the communities that are in blue. And so even though we see free living and particle associated differences, we're also seeing a big difference between the green and red, so the lower and middle river samples, and what are classified as the upper river samples, those that are above the Missouri River confluence. One of the things that we think might be going on is that this is associated to a large increase in nitrate and phosphate that comes in right about this point. So on the left graph is a nitrate in concentrations of micrograms per liter, and each one of these sites is in geographical order along the x-axis. The same thing is true for the phosphate on the right. And what you can see is there's a large spike of nitrate right towards the end of the upper Mississippi River and near where the Missouri River is coming in. It starts to spike slightly above that point, but we do see a large increase in nitrate at that point. And then it sort of levels out in the lower Mississippi River, but it's still much higher than it ever was in the upper Mississippi River in the earlier stages. The same thing is true with phosphate, where the phosphate begins to spike, and then that is carried forward further on down the line. And so you can see that the phosphate concentrations in the red are much higher than they were at the beginning in the headwaters of the blue of the Mississippi River. We can compare these variables with those communities. So if we go back and we look at the free living and the particle associated communities by themselves, and then what we do is we do a statistical analysis where we test for how the effect of these concentrations of nutrients or other variables that we measured are influencing the points on these graphs. We can see that nitrate, nitrite, and phosphate, which are in the left graph in the upper right-hand corner, and in the right graph, sort of in the upper right-hand corner, excuse me, and same thing in the right graph. These are variables that are on long lines, which means they have significant strain or significant impact on what is pulling these communities apart. And so we think that right now, one of our working hypotheses is that the influence of these nutrients at around the confluence of the Missouri, slightly above it, but continuing forward down the river is one of the things that's driving the separation in these communities. Yeah, there's a comment here about large change in dissolved solids and particulate material, and that's absolutely true. One of the things you can see here, we have it in the lower left-hand corner, is Secchi. So this indicates turbidity, and turbidity is also a major driver. We see a vast increase in turbidity changing when we come across that confluence as well. And that's another piece of data that we're following up on. Okay, so if we go back to this, we can kind of look. This is just an overview of the sample points with the blue and the green and the red, and then this delineation between the lower and the upper river. And it looks to us that really the classical middle river, where the Missouri comes in between the Missouri and the Ohio, is actually a lot more similar to the points down below it from a microbial community perspective than the points above it. We're following up with this with some additional statistical analyses. And let's see, and so continuing forward, yeah, so here's the Missouri. And so I just wanted to point out, so that's where that confluence is happening, and that's where we're seeing a big difference between our points between the lower and the upper. And for those of you who are involved in a little bit more advanced analyses like these, we have statistical significance on all this data, and we've got pretty solid R-squared values for this. So we think this is a real effect, and we're doing some additional analyses to follow up on this. Okay. So conclusions, I just want to, towards the end here, I'm basically at the end. I just want to come back to some of these questions we wanted to start with. Are microorganisms in all parts of the river the same, or do they change? Well, they definitely change. We saw that in the bar plots, but we really see it in these NMDSs, where we see that the communities are slightly different. We have additional data showing that particular individual organisms are different in abundance between the top and the bottom. If they are different in different parts, why? Well, we don't know for sure, but based on our analysis, we see that nitrate and phosphate are likely influences of this change. Do the changing influences alter the community? They sure do, and that should say Missouri River. I apologize for the mistake. Mostly at the tributary where the Missouri River comes in, and that seems to be a big change. What are their roles? We do not know right now. This is going to be a subject of some future inquiry, and we definitely are not convinced of which ones are involved in which parts of the carbon cycle just yet, but we have ways of dealing with that kind of information and figuring those things out as we move forward. So I want to conclude by acknowledging the team that has worked on this very hard. Mike Henson, a graduate student in my lab, has processed the samples and run the statistical analysis. Marcus, Pat, Greg, and Jordan were the team that came to us with this amazing adventure and worked very hard to do the sampling. They also did an incredible education and outreach effort all along the river, visiting dozens of schools and interacting with hundreds of people in schools. Fritz Starr was at the University of Washington helping design the study, and the Department of Oceanography there helped with some support in nutrient analysis. And we also want to say thank you to all of the teachers and students that were participants along the way in this study. And so I thank you for your attention and the opportunity to share some of this data with you, and hopefully we can chat again in the future as we get a little bit more from this.
Well, thank you, Cameron. I see there is a question there. What can we look forward to by continuing this experiment over several years?
Yeah, well, I think the saying is that good science creates new questions. And one of the things that we're starting to wonder about is what is the effect on carbon cycling when we see this change in communities after the Missouri River confluence? Does this mean that there is a greater capacity for fixing CO2? Does this mean that there's a greater release of CO2? These are things that we can think about measuring in the future. So actually measuring the way that carbon is used at different stages of the river. The other thing to keep in mind is that all these samples were taken from the surface of the river, and it would be interesting to find out how similar or different things are as we go deeper into the river at certain places. Some places the river is hundreds of feet deep, and there was a recent paper, there's been several papers on the influence of organisms that live in the sediment of the Mississippi River and how they interact with these cycles. And so it would be nice to know how our organisms in the water column itself are interacting potentially with sediment organisms or with surface organisms. And so I think some of the plans that we're starting to think about involve these measurements of directly measuring rates of change of different compounds and sampling in depth, and literally more in depth inside the river itself.
Okay, well, again, I thank you. Very interesting, and looking forward to the follow-up and continuing from this. I just see there's a few people doing a bit of typing. Are your composition measurements based on both richness and, not sure with that one, evenness? There we go.
Yeah, the NMDS plots that I was showing you was just based on relative abundances of those particular communities. We have some additional analyses that are looking at variations in richness and evenness as well, but for the time being, the ones that you saw there are just based on relative abundances of the communities, which I guess it does take into account richness inherently because of the fact that some of those samples won't have some of the organisms that the other samples do, and they'll have different numbers of organisms that have different relative abundances. Yeah. I hope that answers your question, Claire.
All right, it looks like, yeah, and I see Justin is trying to type as well. Hey.
Okay.
I want to thank you again, Cameron, or Dr. Thrush, for your great information. I always want to thank the gentlemen at Ore Northwest, who are just fantastic people to work with, and everybody else. And looking forward to future presentations by yourself.
Great. Thank you very much for having me, and thanks a lot for everybody who was participating.
All right, enjoy. Thank you, and have a great day.